Hello everyone. This week in the lab we're going to discuss microbes that can cause cavities. So we're going to talk about microbial flora of the mouth, and specifically we're going to talk about those that put you at risk of getting cavities. Now, the microbes that actually do lead to tooth decay, we refer to them as karyogenic bacteria. And those are the ones, as I said, can cause tooth decay or dental caries, which are basically cavities. What are some characteristics about these karyogenic bacteria? Well, they produce a capsule. And if you recall, we said that, capsules, those like slimy glycocalyx layers that the bacteria would have, it would help them stick to surfaces. So they would have a capsule, and that would help them stick to your teeth. And then another thing is they're going to be able to ferment sugar. And when they ferment sugar, what is the product that they are going to produce? We've discussed fermentation earlier, and we said that this is going to produce acids. So when bacteria goes ahead and ferments sugar, the main product that comes out is basically acid. So why is that bad? If this acid is now being released in your mouth, well, it's going to start dissolving your tooth. It's going to start breaking down the enamel, and therefore put you at risk of developing a tooth decay. And so therefore, it's super important that we definitely take care of our teeth, by brushing our teeth regularly, especially before going to bed, because you will notice that if you forgot to brush your teeth at night, next day you wake up, you're going to find like a lot of white, right, like plaque, kind of. And this is what turns into plaque eventually, but you find like this kind of whitish layer on your teeth, and this is basically bacteria, right? This is basically kind of bacteria that had the opportunity, right, to go ahead and grow on your teeth, especially also because you didn't brush your teeth. So at night, when we have our mouth closed, etc., the bacteria can get to work. And so by dissolving, and breaking down the sugars that, you have in your mouth, so let's say from leftover, food that you've just eaten, it will go ahead and produce acid. And again, that's going to break down and dissolve like the enamel and put you at risk of developing tooth cavities. So the sugar sucrose, and sucrose, I mean, it's in a lot of food that we eat. Sucrose, that sugar, is actually made up of two sugars. So sucrose is a disaccharide. It's made up of glucose and fructose. Now, there are two Streptococcus species of bacteria that can lead to tooth decay. One of them is called Streptococcus salivarius, and the other one is called Streptococcus mutans. Streptococcus salivarius uses glucose, so when it breaks down sucrose, and now we have glucose and fructose. When that happens, it uses the glucose and it ferments it, and it goes ahead and produces the acid. And then it uses the fructose to go ahead and help it actually form its capsule. So Streptococcus mutans is the opposite. It uses glucose to help it actually build its capsule, because capsule, you guys, it's that it's made up of sugar. So the capsule itself, component-wise of the capsule, it is made up of sugar. So Streptococcus mutans uses glucose to build its capsule, and it uses fructose to go ahead and ferment it. Okay, so you can see it's kind of like opposites. Streptococcus salivarius ferments glucose, while Streptococcus mutans ferments the fructose. Streptococcus salivarius uses fructose to make its capsule, while mutans uses glucose to make its capsule. And then let me kind of show you guys like how do we do the lab so that we can figure out whether you are at risk of developing tooth cavities or not. So we usually ask you to go ahead and kind of like spit in a cup. so that we can collect your saliva. And we collect about 1.5 mils of saliva in like a little Dixie cup. We usually to stimulate your saliva production, you can start chewing gum. So start chewing gum for for a few minutes so that you can kind of stimulate those salivary glands of yours. And then you would go ahead and kind of like collect some of your saliva. And then we go ahead and we dilute it. So we transfer like 1 ml of the saliva into like a 99 ml sterile saline solution. So that's like a 1/100 dilution. And then of course, like you have to shake it and mix it. And then we go ahead and we transfer like, 0.1 mil of that diluted saliva. And then you go ahead and you spread it onto a sucrose plate. So this plate has sucrose. And the reason why, again, we are putting sucrose on this plate because we want to know, right, if you do have the type of bacteria that we just talked about that can break down sucrose and therefore use it to start fermenting, either the glucose part or the fructose, or use the other sugar to go ahead and produce the capsule. Therefore, we have to have sucrose on this plate. And so you would go ahead and you would spread it on this plate, and then of course you would incubate it. And the sucrose plate is actually a differential plate. It's going to help us figure out the colonies of bacteria that we're able to produce capsules or not. So the colonies, these ones right here, you guys, like these large white colonies that you are seeing, and when you zoom in on them, you're going to notice that they are a bit like slimy, moist look, kind of glistening look. Those are the ones that do produce capsules. And so therefore, if you have actually a lot of these large white colonies, then you are more at risk of developing tooth cavities because you have those bacteria that have the capability of basically like sticking on your teeth, right? And that can lead to cavities. So the more you have of these large white ones, the more at risk of you developing tooth cavities. The rest of those bacteria that you have, I mean, those are other bacteria that you have in your saliva, but notice how they don't have that kind of like large white glistening look to them. So those are not, going to produce a capsule. These are the ones that have capsules. And as I said, the more you have of them, the more at risk you are of developing tooth cavities. Over here, let me kind of show you what else do we do. So we go ahead and we, ask students to collect, again, some of their saliva in a cup, and then we go ahead and we transfer 0.2 mils of saliva into these tubes that do have agar, okay, in them, and we refer to them as the Snyder agar, and the media is green in color, okay, so those Snyder agar molten, agar tubes, they are green in color, and since it has agar, we actually do keep them like in a hot water bath to keep the agar melted, okay? And then so you go ahead and you actually transfer some of your saliva into these tubes, and 0.2 mils, if you recall, what is 0.2 mils in terms of microliters? You multiply by 1,000. So 0.2 mils is the same thing as saying 200 microliters, right? Because again, you're going to move it like, you're going to move that decimal 3 spots. So it's 200 microliters. So if you want to collect 200 microliters from your saliva, you can go ahead and use a micropipetter. And so that micropipetter right here, you can use the P1000, and you can set it to 0 to 0, which is basically 200 microliters. And again, 200 microliters is the same thing as saying 0.2 mils. So you can definitely use the micropipetter to go ahead and pull out and transfer out 0.2 mils of saliva, and then you're going to go ahead and add them basically to those Snyder agar, tubes. And then you go ahead and you actually grab those tubes and just roll them between the palms of your hands so that you can mix, like, your saliva into that media. Um, and then you go ahead and you place it actually in the ice bath. Why are we going to place those tubes in the ice bath? Because we want to actually harden the agar. So remember how I told you these two tubes, we keep them in a hot water bath so that we can keep the agar melted until you go ahead and transfer your saliva into them. Once you transfer the saliva into them and you've mixed them and everything, we go ahead and we place them in an ice bath so that the agar can go ahead and harden. Now, why are we doing this? What's the whole point of doing this experiment right here? Well, the Snyder agar tubes, they are actually going to help us differentiate and tell us which bacteria, whether we have bacteria in our saliva that can ferment sugar or not. So if it stays green, then it's going to be negative for, for sugar fermentation. If it turns yellow, then it is positive for sugar fermentation. So these tubes are also going to have like sugar in them. So for example, like sucrose. And so if it stays green, it does start out colored green, but if it stays green, then it's negative for sugar fermentation. If it turns yellow, so if it goes from green to yellow, that means we do have bacteria in our saliva that can ferment the sugar. And the thing is, though, we're all going to have bacteria, you guys, in our saliva that will ferment the sugar. But what we're looking for is how long did it take it, right, to go from green to yellow. The faster it turns from green to yellow, the more of that fermented type of bacteria that you have in your mouth, and therefore it will put you more at risk of developing tooth cavities. So the timing of the color change, that's what really matters. But for all of us, is it going to go from green to yellow? Yes, because, all of us will have bacteria in our mouth that has the capability of fermenting sugars. It's just the timing of it. Okay, the faster those tubes turn to yellow in color, then that means you are more at risk of developing cavities. So let me kind of show you some of the results. Okay, so reading your Snyder agar. So remember, again, it starts green, and then we want to see when is it going to turn yellow. So we have to actually kind of like read the results within like 72 hours. So this is a time sensitive test. And so within 3 days, you must go ahead and actually check on your tubes to see, okay, when is it going to turn yellow? So if your tube goes from that greenish color to this yellowish color within 24 hours, you are at high risk of developing tooth cavities. If it took about like 24 to 48 hours to turn yellow, you're at moderate risk. 48 to 72, you are at low risk of developing tooth cavities. After 72 hours, I mean, that's it. Like, you shouldn't wait, actually. As I said, maximum, you check it, no more than 3 days, okay? After 3 days, after 72 hours, that's it. Negative result, and you will have to go ahead and redo the test if you forgot to go ahead and check on your plates, on your tubes. And so, as I said, like all of us, though, when you do this test, you're eventually going to get this yellow color. It just depends when are you going to see that yellow color. The faster it turns into that yellowish color, the more at risk you are of developing tooth cavities.